Hello everyone. Let's see about organization and expression of immunoglobulin gene of light chain. So first of all, what are immunoglobulins? Immunoglobulins are glycoproteins produced by plasma cells. So these glycoproteins are used by the immune cells to identify and neutralize foreign antigens such as bacteria or viruses. Antibody or immunoglobulin is composed of four protein chains, that is, two heavy chains and two light chains. The heavy chains are larger and more structurally complex, while the light chains are smaller. So, in this video, we will see about light chains. There are two types of light chains, kappa and lambda. The light chains, along with the variable region of the heavy chain, form the antigen binding site of the antibody. This is the region that binds specially to antigens and plays a crucial role in immune response. So in this figure we can see two light chains and two heavy chains, a variable region and a constant region. The At the top we can see antigen binding site, and here we can see the Fab fragment, and below that is Fc fragment. So each light chain protein is made up of 214 amino acids. The immunoglobulin consists of two regions, that is constant region and variable region. So in this light chain, D region is not present. Light chain also have constant region which are found in the stem of the Y-shaped antibody. This region determines the antibody's class or isotype and are involved in various effector function of antibodies such as complement activation or binding to immune cells. The next one is the variable region. The light chain, like the heavy chain, have variable region. These regions are at the tip of the white shaped antibody molecules and are responsible for binding to specific antigens. The variability in the amino acid sequence of the variable region allows antibodies to recognize a wide range of antigens. Next is immunoglobulin gene expression. The first one is VDJ recombination. In B cells, the VD and J gene segments arranged through a process known as VDJ recombination. This rearrangement is catalyzed by RAC1 and RAC2 proteins. But in this light chain, D region is absent. So, during recombination, one V and one J segment are selected and joined together to form the variable region of the immunoglobulin gene. Next is transcription. Once the VJ rearrangement is complete, the rearranged gene is transcribed into messenger RNA, that is mRNA. RNA polymerase II initiates transcription from the promoter region upstream of the rearranged gene segments. Next one is RNA processing. The primary transcript, that is pre-mRNA, undergoes various process steps including 5' capping and polyadenylation to form mature RNA. Introns, that is non-coding regions, are spliced out or removed out, leaving only the exons, that is coding region, in the mature mRNA. Next step is translation. The mature mRNA exits the cell nucleus and enters the cytoplasm. When translation occurs, ribosomes read the mRNA codons in the sets of 3, and each codon codes for a specific amino acids. Transfer RNA or tRNA molecules bring the corresponding amino acids to the ribosome, and the amino acids are joined together to form a polypeptide chain. Next is post-translational modification. The polypeptide chain undergoes various post-translational modification, including folding into its three-dimensional structure and adding chemical group like glycosylation. These modifications result in the formation of complete immunoglobulin proteins. Next is antibody assembly. In the endoplasmic reticulum and Golgi apparatus, the individual immunoglobulin heavy and light chain come together to form a complete antibody molecules. So in this figure, we can see the immunoglobulin gene expression steps. Types of light chain, there are two types of light chain as we already said, kappa chain and lambda chain. Each antibody molecule consists two identical light chains and they can either be kappa or lambda but not a mix of both. The specific type of light chain used can vary different between different antibodies. So first, let's see about kappa light chain. It is encoded by the immunoglobulin kappa locus of 2p11 region of chromosome 2, comprising 40 variable genes and 5 joint genes and one constant region. And a lambda light chain, it is encoded by the immunoglobulin lambda locus of 22q region of the chromosome number 22, comprising 31 V genes and 4 J genes and 7 constant genes, which includes pseudogenes. The region of the messenger RNA or mRNA molecules that precedes the coding sequence of a gene is called leader sequence. This region is also known as 5' prime untranslated region of the mRNA. So this leader region will always be present in front of variable region. Next is RSS, recombination signal sequence. Recombination signal sequence are conserved sequence of non-coding DNA that are recognized by RAC1 and RAC2 enzyme complex during VJ recombination in immature B cells and T cells. Recombination signal sequence guides the enzyme complex to the B, D and J gene segments that undergo recombination during the formation of the heavy and light chain variable regions in T cell receptors and immunoglobulin molecules. So this V gene segment of light chain has 3' end recombination signal sequence, and in J segment at 5' recombination signal sequence is linked. So each RSS consists of a monomer and a monomer, heptamer and intervening portion. So in this figure we can see heptamer, the middle region is the intervening portion, and next is nonomer. So nonomer consists of 9 base pairs and heptamer consists of 7 base pairs, and the intervening portion, either it is 1 turn that is 12 base pairs or 2 turn that is 23 base pairs. Heptamer is a palindrome sequence and nonomer is a ATP rich region, that is adenine thymine rich region. There are two types of enzymes, RAC1 and RAC2. RAC means recombinant activating genes. RAC1 and RAC2 work together to recognize specific DNA sequence called recombination signal sequence, that is RSS, adjacent to VDJ gene segments. RAC1 and RAC2 proteins bind to the RSS sequence in a specific manner, bringing them into close proximity. Next step is synapses. Once bound to the RSS, RAC1 and RAC2 promotes the synapses of two different gene segments. This brings the coding sequence together for recombination. RAC2 stabilizes the synaptic complex, allowing RAC1 to introduce double-stranded breaks at the border between the gene segment and the RSS. The double-stranded breaks create free end that can be joined in various combinations during the subsequent DNA repair process. DNA repair and diversity. The DSPs generated by RAC protein trigger DNA repair pathway such as non-homologous end joining NHEJ. NHEJ rejoins the broken DNA ends, often leading to the deletion of intervening DNA sequence and the fusion of V, D, and J segments. This process results in the assembly of functional VDJ exons, which encodes a part of the antigen receptor. The combinatorial diversity generated through this mechanism allows for the vast array of antigen receptors necessary for immune system function. Next step is p-nucleotide addition. P-nucleotide are random nucleotides that are added at the junction between the rearranged V, D, and J segments during the recombination process. These p-nucleotides are called palindromic because they are typically derived from the sequence that read the same backward and forward, contributing to the diversity, the antigens, antibodies, antigen binding site. Terminal deoxynucleotidal transferase. So this TDT is responsible for the addition of P nucleotide. TDT is a DNA polymerase enzyme that is unique in its ability to add nucleotides without requiring a template strand. This means it can add nucleotides randomly, leading to the introduction of non-template dependent P nucleotides. Diversity and affinity maturation. The random addition of P nucleotide contributes to the diversity of the antibody. This diversity is crucial for the immune system to recognize a wide range of antigen. During an immune response, the B cells that produce antibody with higher affinity for the specific antigen are selectively favored and undergo affinity maturation. This process involves additional somatic hypermutation, which introduces more variations in the variable chain of the antibodies. Next is class switching. Class switching is also known as isotype switching or class switch recombination (CSR). It's a process that occurs in B cells and involves changing the type of immunoglobulin heavy chain while retaining the same antigen specificity in the light chain. The process is crucial for generating different classes of isotopes of antibodies such as IgM, IgG, IgA, IgE while preserving the specificity acquired during VJ recombination. Class switching does not directly affect the light chain, it primarily involves the heavy chain of the antibody molecules. First one is initial IgM production. When a B cell first encounters an antigen, it undergoes VDJ recombination to produce IgM antibodies with a specific antigen binding site, that is the variable region. IgM is the default class of antibodies produced early in an immune response. Next is activation and class switching. When the B cell is exposed to a specific cytokines, particularly interleukin 4 and interleukin 5 and interleukin 6, it can undergo class switching. Class switching is initiated by enzymatic process that recombine the DNA segments encoding the constant region of the heavy chain. The B cell can switch to producing IgG, IgA or IgE antibody depending upon the specific signal received. Resulting antibody class depending on the cytokine signals received. The DNA recombination events, the B cell can switch to produce IgG, the IgA or IgE antibodies. Each of these antibody classes have distinct effector function and it's better suited for different types of immune response. For example, IgG is involved in long-term immunity, IgA in mucosal immunity and IgE in allergic response. Preservation and antigen specificity. Importantly, class switching retains the same antigen specificity acquired during VDJ recombination in the variable region of the heavy and the light chain. Only the constant region of the heavy chain is altered, allowing the B cell to produce antibodies of different classes that recognize the same antigen. Thank you.